Gonna get this started. I'm my own moderator, so, everybody get ready for a 2-hour lecture.
On what?
I'll make it up as I go.
Okay,
Ask questions and
fair enough.
yeah, so time for an afternoon nap. I just kind of have a very general talk here, to kind of introduce, some ideas that I have and, the general group has, but, also give you just a brief intro into some of the things that I do, so everybody's aware. Froze up already? All right, we're good. Something. So, this is actually a slide that I use in a sediment geochemistry class that I teach. And I think one of the most important things here to remember when we think about this in the context of how do we model this throughout the world is that the deep ocean benthic habitat is the largest one on the planet, right? And this is not accounting for mid-ocean ridges. This is not accounting for estuaries. This is not accounting for permeable sediments on the shelf. This is just the deep sea benthic environment. So when we think about how this scales, I mean, this is the most important, potentially the most important environment on the planet, especially for carbon storage, because it is a net carbon sink. And of course, there's all the other things that go along with that. The variability in biodiversity and productivity and how they vary with depth, whether there's light exposure when we get close to the coast, what's the temperature of those locations, the sediment grain size, the abundance of all the different microflora and bacteria and the larger biota that inhabit those environments from time to time. There's a lot of things that we can consider, right, to figure out how to fit this into global modeling. All right, a little bit about what motivates me. I am a coastal biogeochemist, first and foremost. I like working at these interfaces between terrestrial and marine systems. I work in coastal estuaries, really interested in places where the water column has some amount of light all the way to the bottom, because you get these really interesting gradients. Sitting sort of right here between the open ocean, river inputs, terrestrial inputs, it's just a really fascinating and dynamic place to work. And if we kind of take one small snapshot, sort of a 1D look at that, we have gradients in nutrients, commonly very low in the water column, much higher in sediment. Air-sea exchange is very important in these shallow coastal environments, right? The surface area to volume there is very high, so you can have this very rapid exchange with the atmosphere. We have really interesting gradients in flow. I do a lot of turbulence measurements, benthic boundary layer work, so that's an important one that I incorporate into what I do. Oxygen is an obvious one that we all think about in some contexts, and I use that as a primary tracer in the ocean. And when you have light that makes it all the way to the bottom, you get these really dynamic environments with seagrasses, corals, microalgae, all sorts of different things. And then, of course, there's stuff going on in the water column here too, right? So this is kind of like a microcosm of the ocean that I can access all those different boundary layers of study. So more motivation, charismatic photosynthesizers is what I like to do, despite the charismatic megafauna that's in this picture. There's a dolphin that was nice enough to pose with some of the instrumentation. I like these environments where there's a lot going on, where photosynthesis is driving a lot of the carbon exchange. I think you were there on this trip, right, David?
Oh, yeah, yeah.
So I work in tropical seagrass beds. I work a lot in temperate eelgrass beds around here. And I also love to go to coral reef ecosystems. It makes for great pictures for presentations too. So I do a lot of technology development at Woods Hole. So developing new sensor systems, new platforms, different ways to make the measurement of fluxes. It's kind of a range of different things. We have an underwater mass spectrometer now, which is pretty fun, trying to plug in with some of these boundary layer techniques. Gradient flux system, which I'll mention briefly. We're developing automated water incubators to do part of production of the water column. Spectral radiometers that go along with that. Measurement systems for measuring air-sea exchange in coastal environments, where our quadratic wind relationship that we use in the open ocean just completely breaks down. We have to directly measure those fluxes. Fun things like automated bubble traps to look at bubble fluxes. So just to give you a flavor of sort of the technology development side of what I do. And I'll talk today about very briefly about these eddy covariance systems. So this is like hardcore into the benthic boundary layer, right at the sediment interface. And this is a cool video from Matt Reidenbach. You know, there's corals here, there's fluorescent dye, but what I really like to look at, concentrate on is, you know, taking measurements in the boundary layer where you have turbulence interacting with the static boundary. You get these beautiful little eddies. These aren't big ocean surface eddies, right? These are on the side at a very, very fine scale on the order of centimeters to meters. So this is turbulence. And what we do in the case of an eddy covariance system is we're just looking at the covariance statistic between vertical water transport, so how fast is water going up and down, and a scalar, in this case, oxygen. So it's not super complicated. It's simply a covariance statistic. And you take measurements up in the water column where you're measuring that up and down exchange, and then also a tracer along with it. And you can directly calculate a flux from that. So you get both concentration data as well as turbulent fluxes. A sort of related method to that is a gradient exchange method, working with the same background sort of physical principles, but now you're measuring a concentration gradient throughout the water column. And either doing turbulence measurements along with that, or getting profiles of near bottom flow, to which you can also calculate a flux by a couple different methods. We can do this at the ARC interface. There was a picture on a previous slide about that. So we can do boundary layer fluxes, whether it's at the atmosphere interface or at the bottom boundary layer. We have different ways for measuring both metabolism and calcification by incorporating other sensor sets into this. And you know, I just find the technology and engineering side of all this to be particularly rewarding. So just to give you a quick idea of what some of the data looks like. These are a couple different eddy covariance systems deployed in sand flat, in a seagrass bed, and a reef in the Florida Keys. So this is now using hydrogen ions as a tracer, which is kind of an interesting idea that we're toying with now. And just beginning to understand what the flux of a non-conservative tracer is. You know, comparatively, oxygen is pretty conservative, and when you think about hydrogen ions and carbonate chemistry, it gets a little bit interesting to think about what that actually means. And this is an example from 0 to 24 hours. So here we have oxygen flux in the top two panels. We have the hydrogen ion flux in the bottom panels, and you can see that they're across 24 hours of the day. So these are a whole bunch of days averaged together to get sort of a diel curve of what's going on here. So I measure fluxes at extremely high rates. These have hourly resolution. So we can look at diel processes. This is a photic system, right? So there's a lot of photosynthesizers there, and we can really begin to drill into what some of the short-term drivers of the fluxes are, as well as the longer-term drivers as we get more and more data. In the two figures on the left, that's just an accumulation of the fluxes over that time period. And if you look at the oxygen on the top left, see in the seagrass bed in green, we get exactly what you would expect, that net oxygen production. You know, we know carbon, especially these tropical seagrasses, are very good at storing carbon in the sediments, so that's exactly what we would expect. Interestingly, in the sand environment, that was also net autotrophic. There was a lot of microalgae growing on the sands there. You can kind of see the greenish coloration spread throughout the bed forms there. And then you look at the gray, and that's a reef, right? And you're like, whoa, what's going on at this coral reef? And the answer to that, it's obviously very strongly net heterotrophic. It's a big oxygen sink. The answer to that is this isn't really a coral reef, right? This is the Florida Keys, high amount of soft corals, high amount of algae, and are really a degraded reef with maybe 1% coral cover right now, what we think of as typical hard coral cover. So this is really a degraded reef. And if you look at the hydrogen ion flux, it's kind of the same story here, right? So seagrass bed in green is a sink for hydrogen ions, right? Makes sense about what we know about carbon storage there. Sand environment's an interesting one. I've had a couple of these discussions about, you know, you're trying to measure this little tiny net variability. Right? That's the goal of the flux, is we have this big primary production, this big respiration signal, and we're trying to measure what the net is, which is that little sort of 1% in the middle. And what we see in the sand environment is interesting because it is a net source of acidity, right? So it's kind of an interesting result. We're not entirely sure what's going on there, but it's probably related to the high rates of photosynthesis and some interaction with dissolution in the sediments. And then you look at the reef again here, we have a reef that is a very strong source of hydrogen ions, which is probably driven by that massive heterotrophy, that massive respiration that's being caused by the degraded coral reef. So just to give you a quick look at what some of this data is. You know, and the goal here is what is currently done in the terrestrial boundary layer, right? All of these dots on the globe here are terrestrial eddy covariance systems, measuring the flux of carbon dioxide, water, and heat between the biosphere and the atmosphere above it. And so, you know, that's great. I can do a day-long measurement of eddy covariance data, but really the goal is to get to this continuous flux data so we can begin to parameterize things through both space and time, right? So these are continuously operating and they're spread throughout the globe. And this is kind of a long-term goal for me, is to get somewhere to where we can enable that. And that's kind of what this buoy is here. This is a solar powered buoy that is supporting an underwater eddy covariance system that's real-time connected, transmits data back. And this was a 6-month deployment of a long-term eddy covariance system. And I won't get into the data much deeper, but this is a photosynthesis to irradiance curve. So we have light on the bottom, we have oxygen flux on this side. And if you just follow sort of the colored lines and the dates, we have May, June, July, August, peak temperature, still high light. September, the light's starting to decrease, and then October there in the purple. You look at the PI curves on the other side. This is, sorry, not PI curve. This is like that hour of day average that I showed you with the previous slide in the sand, seagrass, and coral reef system. This is in a temperate estuary just off of Woods Hole in a seagrass bed. Again, if you kind of go by hour of day and oxygen flux and follow it up by month, we have May, June, July, our peak all the way up in August, coming back down in September and October. So now that we have this massive amount of data, we're able to average it down, and we can really get really detailed pictures of what sort of that average flux in the system is. So, you know, how do we begin to integrate some of this data with models? And I'll just kind of go through a few slides here. You know, how do we integrate those observations with the models? And we need to sort of figure out how to meet in the middle, right? We need to talk a little bit more and figure out where we need to be. So in terms of modeling, you know, right now we don't have a ton of levers to pull, but how can we put some of these simple ones like sediment type, organic matter, temperature, and depth? What are the other critical factors here that we can integrate to really improve the models? And then how do we make those observations, right? So it's the where, the what, and the when. This is a figure that Chris showed earlier in the day that shows the sort of lack of measurements in the global south. How do we improve that? So this is kind of where we are right now. Where do we want to get to? So I think this where and when question is really interesting. If we think about how things vary globally, this is a very simplified picture of variability, but it's really important, right? So tropical, you know, it's about the same throughout the year. Go north temperate, we have these different maximums related to length of day, nutrient inputs, all sorts of other things. We go to the north polar, we have this spike in the middle. So, you know, if you're out here measuring here and here, you know, there's a lot of variability that you're missing in some of those environments. So sort of the graph on the lower right is an example of sort of long-term sediment trap deployments. And you can see how sometimes they capture some of that variability, some years they don't. So not only do we have the seasonal dynamic, we have this interannual dynamic, and how do we best design observational systems so that we're capturing all the variability both in space and in time. So, you know, thinking a little bit about the different benthic observations that we can do, and, you know, the question that comes here that we kind of developed in a larger model proposal that a lot of people in this room were part of. You know, we thought about what are the most useful, the most accessible, and the most scalable. So, you know, here's kind of a classic sediment trap deployment where you need a big ship to go put all this out. You know, you can collect sediment cores from a whole bunch of different ways. You can sample it for isotopes, pore water. You can take microsensor profiling of that to understand the benthic flux. And then all the way on that other side is kind of what I do, which is sort of complicated instrumentation that probably isn't the most useful, accessible, or scalable. Right? Hopefully we get there in the future, but I think in the short term, we need something a little bit more of the middle of the road in interest of sort of, you know, what is the handshake between observations and modeling? So, like I said, you know, I hope this is where we get to go eventually. That's a detailed data from a long-term deployment from what's that, May to the nor'easter that came in 2021 and destroyed this instrumentation. But you know, 6 months is great, so we're getting there. I hope to get here in the future where we can do this space, we can get to the global south, we can do lots of depths, we can do it in time, we can catch seasonal things, we can capture stochastic events that happen. We can begin to move closer to the coast and do vegetation, whether it's mangrove, marsh, seagrass systems, kelp systems, whatever it is. You know, we want to get to carbon, obviously, and we want to understand the overall carbonate chemistry in the ocean. And the way to get there is technologies, new sensors, new platforms, but that's really going to be a longer road. So this is kind of a process from that proposal that I mentioned that a lot of people in this room worked on. So I'm definitely taking credit for all of their hard labor on this. But, you know, thinking a little bit in a flow diagram what this looks like. So, you know, can we go to the global south and make observation cross-shelf transects? What is the sort of baseline data that we can collect on cruises of opportunity, right? And there's a lot of details in here, but you know, we want to get to the basic model parameters that are defined by these observations. And kind of building this cohesive data set that actually does what the model needs. So there was a lot of back and forth here figuring out how do we really bring all this together so that we can implement the model, we can figure out the carbon budgets, but also transfer sort of the knowledge that we have on how to measure fluxes to other people. So it's more accessible so we can spread these measurements out throughout the globe. And sort of like the next exercise we did is, okay, how do we do those observations and what does it look like? And this kind of worked in this framework for this proposal, but you know, thinking about different tiers of measurements and what can everybody do? What do we think that most people across the world could be able to do? And it comes down to, you know, physical parameters like temperature and salinity, and then thinking pH and oxygen might be pretty accessible. You know, simple measurements of fluxes from core incubations or other techniques that might have available, and some basic sediment parameters like porosity, density, basic loss on ignition, carbon content, and wet and dry biomass. And we can get more complicated. We can start to add things like inorganic nutrients, DIC, thinking a little bit about dissolved gas fluxes, and then getting into carbon nitrogen ratios. And then, you know, tier 3, maybe we only do this in a couple of spots, but then thinking about all the more detailed chemical measurements that we can do. Maybe we do some of my work there when we finally get to tier 3, and then more detailed analysis in the sediment. So, you know, then there's all these other processes that we have to think about, right? So how do we figure and model this all out? I'm not going to go through this slide. Everybody's got a box on here that they can identify with, but you know, that's why we're all here is how do we make all this okay? I think I'm about done. Since I'm self-moderating, I can go multiple minutes over, but of course, I'd like to thank everyone in my lab and all the fun things we develop and all the fun we have in the field.
Start off with a pretty rendering. So I, for the very first time, have met most of you, and we don't have too much in common, but we all love sediments. And so I'm coming at it from a perspective generally of the Navy. They are interested in characterizing the environment so that they can better navigate and assess threats and et cetera. But I really like the idea of taking those techniques and inverting them, right? So using acoustics to invert for properties of interest to other communities. So my title is New Methods Scaling Up Data in Time and Space. And I put quotations on new because they're not super new. And the data has been taken is on Amazon Web Services and is just sitting. So I'm using these techniques to get something useful out of it. And this is just a rendering of some fine-grained sediment. We did a lot of acoustic measurements off the continental shelf. So. Okay, so here's an outline of the talk. Since you don't know what I do, I'll just give a few brief overviews of some of the research projects in our lab. And you'll see these faces scattered everywhere. You know, I always love to collaborate across disciplines. This is an example of that, hopefully. And so then we'll start talking about, you know, probably why I'm here. And it's this link that I found between sediment, organic matter, carbon, and acoustic properties. So I'll talk about some of those measurements. And then how we can use that relationship to do a geoacoustic inversion using a physics-based model to do some scalable seabed characterization. And then we'll talk about potential collaborations. So I do work in seagrasses as well, in the lab, in the field. This is just the project we have in the lab where I'm working with a fluid dynamicist and we're imparting monami, this sort of turbulent structure that occurs on top of wheat fields and seagrasses. And we're trying to correlate the acoustic field with the surface expression of the water. There's my master's student, Langdon and Allison. Langdon just defended last week. Here's some of his results. We're basically taking a physics-based bubbly acoustics model and we're inverting for void fraction of air in these seagrasses that have been seeded with bubbles in the lab. And you can see the changes in these void fractions as the seagrasses expand and contract. This is brand new. We're just talking about it now. We just got a little bit of seed money to do this really cool acoustics technique called ambient sound correlation. Probably the coolest application I've seen is this one here, published in 2015. And they have these two cabled acoustic arrays that are, I don't know, 100 kilometers apart from each other. And using the ambient sounds from polar calving events, so glaciers sort of breaking off and making low frequency noise. This noise travels across the ocean. And if you average enough times in this sort of interesting signal processing methodology, you can do time of flight measurements across hundreds of kilometers. So you get an average of sound speed and therefore temperature. So you can do large scale ocean temperature monitoring using just ambient means. So we're trying to do that in a seagrass bed in the Great Bay Estuary in New Hampshire to see if we can do a low cost, low power application to measure seagrass productivity using the oxygen bubbles that come out of the seagrass. I'm working with the Coastal Response Research Center. I don't know if you all know Nancy Kinner, but she does a lot of work with oil slicks, and I was brought on board to characterize oil slick thickness. These are some of my students as well in the project. And lastly, before we get to the organic stuff, we're basically take echo sounders and just point them at the bottom and leave them there for a long period of time. And we can correlate, you know, how the seabed backscatter changes in time and see how what the response of the seabed is to biological stressors or hydrodynamic stressors. So here, as you see, the mean wave heights over time. So you see this big storm event here cause a pretty drastic change in the backscatter. In the y-axis, you have distance from the echosounder, and then on the x-axis, you have the time that those pings were taken. So you can see the spans 5 months in time. And so we're taking these techniques and we're bringing it to the deep with the ONR, multi-institutional university research initiative, MURI, that's starting this summer, and we're going to the Pacific coast and then seeing how this changes in 4 kilometers of water. Okay. Sort of my main question is, I'm trying to connect sediment microstructure, organics, with sort of bulk sediment acoustic properties. Because that's what acoustics is really doing. It's sensing the substrate. I'm not going to be able to help you with, oxygen going in and out or other gas exchanges, but if whatever happens in a biogeochemical framework, if that changes the sediment in any way, if it changes the porosity, if it changes the permeability, if it changes, all these other parameters that is important to acoustics, then I think, we could have a nice collaboration. So we're studying both properties like shear waves. So I'm working with a biomedical acoustician in University of Pittsburgh, John Cormack, and we're taking the ultrasound probe that we use to see babies in the womb, and we're focusing that acoustic energy in a small point, and it's such a high amplitude that it actually pushes the sediment. And then we're using the same probe, and we're imaging the Doppler, the motion of these particles in time. And it manifests itself in a shear wave that propagates out. So you can actually measure the shear speed of a mud, and relate that to microstructures. So we do X-ray microscopy and backscattering electron microscopy and energy spectrum spectroscopy to try and find where the clay platelets are, where's the organic matter. And we stain the organic matter with various precarious chemicals to see them in the SEM, identify their elemental. I'm trying to answer this question. And I was interested in this. Missed the other question. But I was interested in this because I found this relationship, right, between sediment organics and acoustics. So these are some of the questions, how do organic content affect sediment acoustics? And then the inverse, and then why? This is the part I'm still trying to unravel, is why exactly is there this relationship? Independent of why, this correlation still exists and we can exploit it. We can use it potentially to back out organic carbon, sediment organic carbon in situ using acoustics data. And we'll talk about the latter. So here's the sort of correlation that I found. This is not my work. This is really the only work that I found so far that when they go out and they measure sediment properties, they have both acoustics and organic. In this case, it's organic content or loss on emission. And so I'm trying to build up that database to see if this is non-site specific. And so far, it kind of points to maybe this site here in the green LLM is the Lower Laguna Madre, like a hypersaline lagoon that has a lot of Thalassia tostilium, seagrass and turtle grass in there. And that's where we first found this relationship back in 2019. And we also, this is actually the wrong citation, but anyway, so MB is Mobile Bay, it's a estuary, a lot of different kinds of sediments, that's in blue. And this black line is essentially the same line that's plotted on this paper here, Endler et al. And this is taken in the Baltic. So you can see that they all collapse into a nice sort of curve here. And I thought that was really great because I tried plotting against acoustic parameters against everything else, like mean grain size, mud content, all of that. It was always a cloud, dot cloud. So this idea of this impedance ratio is really the density of the sediment times the speed of sound of that sediment divided by the density of the water times the speed of sound of the water. So it's like a normalized quantity. And you can compare, their locations. You see that there's this really nice region, right, between like 0, which is basically sands up here, all the way down to soft muds, loss of emissions above 10. There seems to be a lot of scatter here. So could be something to do with the type of clay mineral that's there. Or, you know, the amount of organics that's present. And I don't know much about, you know, depending on where you are in depth and how far you're away from the shore, you know, it could have been decomposed before it reaches the bottom. I'm curious to hear your thoughts on it, but it's reasonably sensitive, you know, below 10% loss of emission. So I'm not going to go into this too much, but the idea is that there are generally 3 physics-based sediment acoustics models, and they incorporate various mechanisms by which sound is either attenuated or propagated in the sediment. The point I want to make here is a lot of these inputs are empirically derived using sediment data. So you need to go out and take acoustic data and sediment data and do these fits to get these sort of immeasurable parameters. And part of what I'm trying to do is see if there are any constitutive parts in the sediment that can predict these. Can this be a predictive model or not? But regardless, you know, we should still all of these models, they don't include organics. So the Navy just thinks sediment is just water and grains, and that's it. And I'm trying to relate these. This is currently under investigation. So this is still work in progress, but I'm working on it. Okay. So how can we exploit this relationship? On my own time. Good. Right. So there is actually this time domain scattering model that already exists. This is not new method, no. But it basically takes into account all of the geometry and the transducer properties and the, you know, how far you are from the seabed. And it embeds as this wave is sort of spreading into the sediment volume and on the surface, it's shedding energy back. So it's backscatter. And in the volume, you get a volume backscatter, which is akin to some kind of, you know, heterogeneity that's in the sediment that is basically changes in density, or if there's shell components in there, it will scatter back some. And all of that goes into the model. And so you get the depth, your frequency you're being with, this impedance ratio I was talking about before, interface roughness, volume heterogeneity, and Andrew, right there, my master's student has been coding up this model in Python, and we're trying to make it publicly available for a reason I'll talk about in a second. And this model works really well for sands and muds and clays, not so much for gravels. So these physics-based models use this Kirchhoff approximation that I want to talk about, but it's not valid for those really coarse sediments. Okay, so scaling in space and time, right? There is a massive database, water column sonar database, that's mostly NOAA fisheries going out and counting fish. And they need to count fish and figure out what kind of fish is out there, so they calibrate all of their echo sounders every time. It's really nice for sediment inversion. So this in red is the Northeast Continental Shelf since 1998. They go out multiple times a year. Every cruise they go out, they collect like, I don't know, 50,000 pings. Okay. And they do that for the last couple of decades. And it's just this massive database. And no one is looking at the bottom maps except us. So this is kind of something, this is like called an echogram. As you go on in range, this is the sort of depth from the surface, and you'll see this is the Scanton Basin, and it goes up to Jeffrey's Ledge, you know, somewhere up here. And you can see this signal here is what we're using, this sort of ring. It looks kind of like this. So if you stack some of the things together, the black line is the ensemble average, and the gray is sort of this other, just basically all the things stacked on top of each other. This idea of backscatter is a sort of plastic quantity, so you have to do these sort of 100 pane averages to get something and invert from that. We're using a Bayesian framework, so it's a very honest way to invert for data, because if there's no information content in the data, it will reflect in your posterior probability distribution. So you'll get probability distributions for all of your inverted input parameters, which are these, right? So this is impedance ratio, interface strength, scattering strength, and then this is, you know, a volume parameter. And you're basically sampling the posterior probability distribution using Monte Carlo approaches that satisfy Bayes' theorem. So this is the potential indirect inversion products. I'm trying to be careful here. I'm not trying to say that acoustics is going to solve all your problems. This is quite involved, and we're just starting. And I didn't have my talk uploaded because I was still running our very first inversion result in the room. So you'll get to see it, and I'll sort of try to interpret it with you. But the idea here is these are some of the things that we can get for it. So mean grain size, porosity, both of these mean grain size. Oh, I have a bunch of errors. So mean grain size is usually from this roughness parameter. So the coarser, the higher the backscatter strength in the interface. Porosity is really closely correlated with acoustic impedance. So is organic carbon. This idea of bioturbators. So I just learned that bioturbators aren't just mixers or tube builders. They can bioturbate the chemistry, I suppose. I didn't realize that. But these are sort of things that can cause mixing in the sediment, heterogeneity in the sediment. That comes out of this sort of volume parameter. And this idea of having, you know, temporal and spatial maps. But the kicker is we need a lot of ground truth. And that's part of the other half of the reason why I'm here, is to see, you know, what kind of ground truths you guys are interested in or usually take. Because this model inversion needs some benchmarking. So I'm not going to just believe whatever comes out of this thing. I need to benchmark it first. So we have a lead at USGS. I have a colleague there who's giving us a bunch of qualitative data, like grain size distributions, loss of emission, those kind of things. So this is still work in progress, obviously. So this is a preliminary inversion result in the Standard Basin. So this is Jeffrey's Ledge, and this is Standard Basin. And here are a bunch of ground truth measurements that were taken in 2002. And the first scattering EK60 is the type of equipment that they use, and we're only using that equipment for now, just so that it's easier. But we're trying to get equipment that has been ran back, you know, past 2008. But this was ran in 2008, and you can see this transect essentially manifests itself in this echo thing. So we're going to take a little section down here of this super muddy stuff and do an inversion. And so we run it in this Bayesian framework. We run it like 200,000 times, and it will accept or reject, you know, models depending on the likelihood function that I assign to it. And it samples in parameter space really efficiently and everything. And so we can get this data model agreement. But the kicker is, what are the parameters that cause that best fit model? So this is just the best fit. But we actually ran a bunch of these models, and I haven't done it here because like I said, I just ran this the other day, or just earlier today. But you can essentially get nice confidence intervals as well for the model results, 95% confidence intervals that I will do eventually. But you can also get what's called, I have a much better, maybe it's over here. No. Okay. Well, this was my results that I ran last night, and I continued running them this morning, and they look a lot nicer and smoother, but you can always come to me if you're interested. But the idea here is you get all of these parameters are the inferred parameters essentially. So this down here is the volume scattering coefficient. That's really well resolved. It's a value that's dead in line with literature values. This here, MV, is the mean grain size, which we've parameterized in a funny way, and we've parameterized it this way because it's what's been done in the literature, but we're going to change it. And so this sort of mean grain size relates in this idea of impedance and interface roughness in one, when they're not necessarily perfectly correlated. So that's something we need to do better, essentially. And the idea here is that if you were to take this value of mean grain size, like the best fit value here, the sort of most probable value, and you stick it in this empirical relationship that we resolved to get impedance and interface roughness, the ground truths are about an organic content of about 10% loss of ignition. And that lines up really well with the impedance that corresponds to. Even though the mean grain sizes aren't lining up so well, the impedance is. So we're planning on parameterizing using impedance. There's all these parameters, these relationships that have been published in acoustics literatures that are parameterized off of impedance. And from that, we can get, using some kind of relationship, get organic content. We're in an area that's not the most sensitive area. Like I was saying, anything below 10 is pretty sensitive to changes in impedance and organic content. But up here, we're getting some kind of scatter. So it's a little on the low end of this curve. But that's the basic idea. We have a long way to go. And I'm here just to listen. And everything I learned about biogeochemistry, I probably learned this morning. So it's been fun. Yeah, and looking forward to contributing and seeing how we can overlap and collaborate. Thank you.
Okay, yes, so I'm going to talk about, in the schedule it says sediment water fluxes, but I'm going to focus mostly on trace metals because that's much more my expertise. And I think there's a lot of other work on like oxygen and carbon and other types of fluxes that are super important that I'm not as knowledgeable about. So I am going to focus on that. And I just wanted to go through a little bit about me in case I haven't talked to you yet. I'm at Florida State University and been at Rutgers and Stony Brook, and I would describe myself as a seagoing oceanographer. So I'm very observational, kind of on that side of things, and I focus on sediment and trace metal biogeochemistry. And the reason I'm here and kind of where I'm trying to go with my research is to link the kind of small scale observations that I make to larger scale processes like carbon cycling and climate. And I'm really kind of excited about the things that we're talking about here because I think the way forward is to, number one, coordinate observations, and then also collaborate with modelers from beginning to end of a project. I think that's kind of important, and I'll give an example of that later. So just a little bit of background on why I care about trace metals. So many of these metals are essential for life, and they're called micronutrients because they are required for a number of really important biological functions, but they exist at really, really low concentrations. So that's why they're called micronutrients. And iron is especially important. There's already been a lot of discussion about iron. So I am going to focus on that, but the other ones are important too, just maybe, in more sort of regional ways. And also we don't understand a lot of the kind of interactions between different trace metals in terms of biological limitation. And I will mention that, of course, some metals at high concentration can be toxic, have the opposite effect, but that's not really what I'm talking about here. That's kind of a separate topic, but it could be relevant. I mean, if you're calculating trace metal fluxes in like contaminated sediments, then some of these same methods that I'm going to talk about in a minute could definitely still be relevant, just with a different effect on biology that I'm going to talk about. So since trace metals like iron are required for life but exist at very low concentrations, iron in particular can limit phytoplankton growth in large areas of the world ocean. And note that this is a model result, so there's lots of different ideas about what is limiting where. This is just one idea, but it does illustrate that, the dark red and orange illustrate either strongly or weakly iron limited systems. So this means that the phytoplankton there have enough of everything else they need, nitrate, phosphate, light, whatever else. They just don't have enough iron. So if you add more iron to the system, then that'll increase their growth. And you can see that especially in polar regions, so like the Southern Ocean and parts of the Arctic, iron can be really important because the other macronutrients are plentiful. So this is one of the reasons that I am very interested in sediment fluxes of iron. We really want to know all the different sources to see how iron is behaving as a limiting factor of growth. And sediments are really important reservoirs of trace metals. So I've just illustrated the typical concentration of iron in Earth's crust is like 40,000 ppm, and iron in seawater is like 0.001 to 0.1 ppb. So really, really huge difference. And this is partly why iron from sediment sources, usually from lithogenic particles that are dissolving, is potentially a really important source of iron in the ocean. And we had a couple of beautiful talks about this sediment biogeochemistry and the redox ladder and all that stuff. So I'm gonna skip over this a bit, but I just want to remind you that sediments concentrate these inorganic particles, for example, that are rich in iron, and organic matter. And there's also really rich biological community there. So there's just a lot of reactions that happen in sediments. And one of the most important ones for iron cycling is the reduction of iron oxides during the oxidation of organic carbon by microbes. And this releases a dissolved form of iron that is reduced, and that can either be returned to the overlying water through diffusion or mixing, or advection, depending on your system. I guess mixing is kind of advection, but biomixing is what I was thinking of here. Or under certain conditions, for example, if it's very sulfidic, then iron can be locked up in sulfide minerals, and sediments can be a bit more of sink. So it's a very complex system, and this is part of why iron from sediments and iron fluxes are super difficult, because it's not like there's one process that's happening everywhere. Even whether the sediments are a source or a sink can vary depending on the biogeochemistry. Okay, so okay, sorry. I think we didn't. It's okay. I'll go to the end. I had added a slide right before lunch, and I think that this may be the old version. It's okay.
Oh, sorry.
It's okay.
Restart. So.
Yeah, it's okay. So I guess I'll just say now, and maybe when I get closer to the end, I have it as like an extra slide and I could just move it forward. But I wanted to explain that this kind of way that iron can be released to the overlying water is, we call that a reductive source. So that happens when oxygen is drawn down and the iron is reduced. But a lot of studies of water column iron isotopes, for example, have suggested that non-reductive sources are also really important in providing iron to the water column. So that's a point I'm going to come back to, but I do think it is important because I think most models these days that incorporate benthic fluxes link the iron flux to something like carbon oxidation rate or bottom water oxygen. And that only captures the reductive sources of iron, and it does not really capture the non-reductive sources. So I have some nice figures showing that, but I'll show you that later. Okay, so some of the ways that sediments kind of communicate with overlying water. I think I probably don't need to make this point to this crowd, but the sediments and the overlying water are really an open system. I think in a lot of cases, they're kind of considered as totally separate boxes, but they really are an open communication. And if we're talking about dissolved constituents, the pore water is kind of the key route that stuff can, or the way that stuff can get from the sediments into the overlying water. And so this figure just shows, I'm sure you all know this, but the pore water is basically just the liquid that fills the spaces between the grains. And so this transport through the pore water into the overlying water can occur through diffusion. So just along concentration gradients, the sediments often have higher concentrations of certain things. The overlying water has a lower concentration, so whatever that is gonna move from the high concentration in the pore water to the lower concentration in the overlying water. There can also be advective forces, usually not in clay-rich sediments, but in things like sands. There can be tidal pumping or potentially groundwater flow, different forms of advection. And then finally, bioirrigation can be really important. So worms that, make tubes and then flush their tubes. That can be a really important form of communication between sediments and the overlying water. And this is just a picture of a core where I was trying to do an incubation, and there was a gigantic bioirrigator in there that was messing up everything. So yeah, so that is the dissolved constituents. And then I also want to point out the particles do also, usually the particles come from the water down to the sediments. Particles sink in water, but there can be resuspension events like a storm or a change in current speed or, any kind of like turbulence over topography. And then also bioturbation, that's just the physical mixing of the sediment, can also provide make particles go back into the bottom water. And the role of particles, if we're focusing on the dissolved, so for example, iron, we focus a lot on the dissolved phase because that's the most bioavailable, that's the easiest to transport. And the effect of particle resuspension on dissolved iron in the water column is a little unclear. In some cases, the particles can, release dissolved iron into the water, but particles can also cause iron to adsorb onto them and kind of strip it out of the bottom water. So it's a little bit unclear how particle fluxes from the sediment surface might affect the dissolved water. Okay, so benthic fluxes are important sources and should definitely be included in models of trace metal and carbon cycling. But there are a number of challenges that make this kind of difficult. So I'm going to go through some of the challenges and try to propose some solutions or, points of somewhere to start from to resolve these challenges. And the challenges kind of fall in three categories that are related to each other: methods, differences in methods, variability. Sediments have a large degree of variability on small scales, and then scalability of whatever observations we make. So the first issue with the methods is that fluxes, and here I'm talking about iron fluxes pretty specifically, so just in case you're wondering. If they're determined with different methods, they're not very intercomparable, unfortunately, because each method has its own assumptions. So I'm going to walk through some of the methods for determining fluxes that are used most commonly. This is the one that I'm most familiar with. And this is using pore water gradients. So basically just measuring the concentration of iron in the pore water over the sediment water interface, and then assuming simple diffusion. And it's a pretty easy calculation. All you need is the sediment porosity and then a diffusion coefficient for the sediment and for the specific parameter that you're measuring, and then the concentration gradient. And then another way to kind of calculate fluxes is by looking at the water column. Could do things like isotope mass balance. There can be some benthic tracers used. So basically trying to quantify like exchange with sediments in the water column. That has some issues that I'll come back to in a minute. And you definitely have to contend with the benthic boundary layer when you're looking at water column analyses. Okay, then benthic chambers and landers are a really big one. This is kind of considered state of the art, I would say, for determining fluxes. And this just shows a diagram. It's basically a chamber that goes and sits on the seafloor for some amount of time and just measures the change in iron or whatever else over time. So it's kind of like an in situ incubation. Then eddy covariance, we heard a little bit about, not as useful for trace metals, but definitely a method, kind of in situ method of measuring fluxes. And then you can do also core incubations, like I mentioned before in the lab, and those have their own issues. Okay, so now I'm gonna go through all of the issues with all of these. So focusing first on the pore water gradients, the reason that I have really focused on these, and I do still, I still kind of like carry a torch for pore water gradients because they're just so easy. I mean, they're not easy, but relative to other methods, it's really the most straightforward way. So I really am focusing on trying to improve how we use pore water gradients. And there's also the largest amount of historical data. So most fluxes in the literature will be based on pore water gradients. However, pore water gradients depend on the pore water data that you collected. So there are a couple of different methods for collecting pore water, and they aren't always perfectly the same. So for example, if you're looking at really anoxic pore waters, you might use something like rhizomes here. These are cylindrical filters that you insert into the sediment core and pull a vacuum and pull out pore water, and that's really good because you can minimize oxidation. But if you don't have that set up, then you might do something like I have here, where you centrifuge the sediment and get the pore water that way. And that's good because you get more pore water, but there are higher chances for oxidation. And it's much harder, I mean, when I've done this, I work in a glove bag and, in a cold room and try and mimic in situ conditions, but there's a lot of room for error there. And it's really easy to get oxidation artifacts. It's also difficult to collect clean samples. And you might think that sediments, like it's impossible to collect clean samples from sediments. But if you are careful, you can see pretty low concentrations in something like iron, and it's obvious when there's contamination. There's, one point that will be way over here. And so you kind of have to stay as clean as possible, try to minimize particles as soon as you've separated the pore water. So anyway, that's another kind of challenge. And then finally, one of the most important ones is the sampling resolution makes a really, really big difference in these fluxes because the concentration gradient plays such a role. And ideally, you have really high resolution. And one way that you can do that is with something like I have used here. It's a planar optode. Basically, it has a chemical bound to it that turns pink when it contacts iron too. So you can basically insert this kind of plastic rectangle into the sediment core, like you can see there, and then get kind of a two-dimensional, really, really high resolution concentration of iron 2+. So this was, we calibrated in the lab, and this is the two-dimensional pore water iron concentrations, and then you can average across the width here and get the pore water profiles. And then you can basically use this gradient, which is, sub-centimeter scale to calculate the flux. But that only works for iron 2. So again, if you're looking at non-reductive sources, and there are other issues with these sensors as well that may not be perfect. Then finally, another really important part is the diffusion coefficient that you use to calculate these fluxes is dependent on the speciation. So usually when we're doing these calculations, we assume that the iron is just iron 2+, and that has a diffusion coefficient that we can get from the literature. But if we're looking at non-reductive sources, for example, we know that the iron in the pore water is not iron 2+, almost certainly, because it hasn't been reduced. And so in that case, it makes most sense to try to calculate a diffusion coefficient based on the diameter of the potential iron particle. And when I say particle, I mean like colloid. So like something that is like you can measure a diameter, but it's not a particle that would settle through water. So I have used this. It's the Stokes-Einstein equation, basically just relating the diameter of the particle to the diffusion. And it makes a really big difference. So this table shows using the same data, same everything. If you assume everything is iron 2+, you might get like 17 micromoles per meter squared per day. If you assume everything is, say, siderophore size, that's a ligand that binds with iron, that's 1 nanometer, that's 11.5 micromoles per meter squared per day. And then if you're assuming it's a large colloid, so like some blobby like organic molecule, or maybe a nanoparticulate iron like ferrihydride, that gets down to 0.07 micromoles per meter squared per day. So like orders of magnitude difference depending on the speciation of the iron. Okay, also, this is a really big topic, but I just threw it in as one bullet point. So bioturbation is really difficult to represent in gradient calculations like this, or these sort of diffusion calculations. A lot of people put basically some kind of factor in the equation to represent bioturbation. And that works, but I think there's a lot of uncertainty about how those, factors are determined. And it's very different for, like, say, different bioturbators, different species and activities. Then finally, the last issue with pore water gradients is it's uncertain how they translate, the fluxes translate to the water column. So there may be unresolved gradients right at the surface. There may be substantial reoxidation or complexation, scavenging in the bottom water. And it's not shown in these calculations how that iron is transported to the surface water, which is where we want it if it's gonna affect productivity. So some issues there. And then I'm just gonna go through these quickly because this is less my forte, but I just want to explain some of the issues. For the water column analysis, it's great because it's an integrated signal, kind of smooths out any spatial variability, and it accounts for those water column processes like scavenging, dissolution, whatever. But it's often not feasible to capture that benthic boundary layer in sampling unless that's like really only your target, which it's usually not when you're studying the water column. And it's also difficult to differentiate processes and rates. So you're kind of looking at like the net result of the flux, but you don't really know maybe what processes contributed to that, which maybe doesn't help with a model quite as much. And then also horizontal affection can be, that's an example of something that can sort of mess up those. Benthic chambers, also in situ, kind of measurement of net exchange, and it includes the effects of macrofauna. But it can, depending on the system you're using, there can be an issue with oxygen drawdown, short incubation time. If the rates are slow enough, you won't really capture the flux, and it doesn't capture some of the water column processes. And possibly most important in my mind is they can be really challenging to deploy and recover, and therefore we have really limited spatial coverage. This was from 2016, so hopefully there's like a lot more now, but at least in 2016, these show all the spots where we actually have benthic chamber fluxes. Okay, then eddy covariance, really great way to measure, gas exchange like oxygen and carbon, but it requires fast response in situ microsensors, which we don't really have for metals. So if you're focusing on reductive release and you want to tie your iron to the oxygen, then you could probably use these systems, but as I've said, that may not be totally accurate. And then finally, incubations in the lab. You can do controlled experiments like adding amendments, adding carbon or something and seeing what happens, but it's really difficult to replicate in situ conditions. Then I'm just going to go through this super quick. Another issue is there's a large degree of variability in the spatial sense, both on the large scale and on the small scale. For example, this was a worm tube on one of these planar sensors, and you can see that in the tube, there's like, it's almost like a second sediment-water interface sort of within. I think this is a filled tube, but initially that would have been, like increasing the surface area for flux to occur across. And then there's temporal variability across seasons. That's super common. So summary of the challenges, there's kind of a trade-off between pore water sampling and accuracy. So, a trade-off between ease of sampling and accuracy. So pore water sampling is great because it's really easy, but it requires a lot of assumptions that you may not be certain about the accuracy of. And benthic chambers are typically considered the most accurate, but are super labor intensive. So what I would suggest is maybe a kind of two-pronged approach to improve the methods, increase the ease of use and access to benthic chambers. It sounds like there's some really cool work happening on this. In my dream world, we could have like a little ROV that's like you know, a little benthic chamber that goes and plants itself and then 2 days later swims over and plants itself somewhere else. That would be amazing. But also I think in the meantime, calibrating the pore water sampling with benthic chambers is worth working on. Trying to get better diffusion coefficients, better representing bioturbation, and see if we can kind of get close enough just using the pore water. Use all data sets with extreme caution, modelers, especially when you're including multiple studies that maybe use different methods. Maybe best to start with regional modeling in areas with good observations, and definitely ask someone if you're not sure, because there's usually a lot that maybe the paper didn't explain a whole lot. I have a bit of an example here. I'm sort of running out of time, but I'll just say really, really quickly, this is a project I'm working on now. In the Amundsen Sea, I basically collected a bunch of sediment cores. Each of these dots is a sediment core, and I calculated a flux. So really using the gradients, really low fluxes, less than 0.1 micromoles per meter squared per day. We assume that this is non-reductive flux. And we incorporated those flux values into a high resolution circulation model, just treating iron as a passive tracer, just seeing if we can replicate the bottom water iron concentration that's observed by putting these fluxes into a model. This was done by Pierre Saint-Laurent at Mincks. And it was really helpful and interesting because the background blue is the model result. And each of the points is an observed bottom water concentration. And you can see that in some places, like the arrow and the yellow circle, the model and observations matched fairly well, but in some places it did not. And so we went and looked at those places and saw that that was where we had seen this phytodetrital fluff on the surface that was also high in manganese and cobalt, suggesting that there may be some rapid decomposition of this fresh algal fluff, consuming oxygen and creating microenvironments that releases iron. So this is just an example, and I'm happy to talk more. I went through this really quickly, but of it, like where we sort of use observations to inform modeling, which then informed our further interpretation of our data and possibly informed like further studies. So yeah, I think I'll stop there, and I'm happy to chat after if anyone has questions.
I'm going to change it up a little bit here again. I'm a biologist. I'm trained as a biological oceanographer, as a oceanographer primarily, and I've worked in polar regions pretty much for my whole career. So my ecosystem dynamics is going to be focused on the systems that I know best, which are the high latitude regions. And in the Arctic in particular, this is a region that's undergoing a lot of rapid change. So big changes in sea ice coverage, and sea ice is an important driver of a lot of other processes, including water mass structure and primary productivity patterns, timing and nature of blooms, where blooms are occurring. So one of the predictions, just to give an example of how we're thinking about this system, of how we might see the system changing going forward, is shifting from what we currently observe as what people call a benthic dominated system, where we have a lot of flux of ice algae and open water phytoplankton actually exported to the seafloor. And that fuels in turn a pretty rich and biomass heavy benthic component to the system. And that's important from the standpoint of coastal communities in this region, because there's a lot of indigenous communities that subsistence hunt and fish for marine mammals that are benthic feeding species. Predictions going forward are that less of that flux or less of that productivity is actually going to end up on the seafloor, and partly because of the nature of the timing changes in the marine dynamics, we'll see more of a partitioning of resources into the pelagic community. So those are really fundamental shifts that are predicted for this region. And the motivation for a lot of the work that I and my colleagues do is to try to predict how these changes are going to be affecting critical functions of the ecosystem, and of course, abundance and condition of a lot of these harvested species that our economy in Alaska is really dependent upon, as are the indigenous communities. So to give you, I know not everybody thinks about the Arctic all the time, so just to give you a little bit of geographical context for the region that I'm talking about. The map here shows sort of the mean current patterns through this system, and there's a lot going on here. So the main points that I want to emphasize are that this is a what's referred to as a shallow inflow shelf. So it's northward advection of currents across this region. It's a shallow, like roughly 50-meter on average shelf depth. And so the mean flow is high nutrient deep water from the Pacific that upwells onto the shelf in the Bering Sea. There's you can see 3 large canyons in that image. There's some smaller canyons as well. They're basically like conduits for this water up onto the shelf, which is part of the reason for the really productive fisheries that we have in the Bering Sea. And then everything gets squeezed through Bering Sea up here. So currents speed up through that zone as they get forced through that narrow opening. And then on the north side, everything kind of slows down, and that leads to some interesting dynamics that influence particle flux, particularly north of this strait. So that's the mean picture. And then another point that I'm going to come back to in some of my data slides is differences between the red current on here is the Alaska coastal current, which is fed by the drainage of a couple of large rivers, including the Yukon River. So a warmer, fresher water mass with a lot of terrestrial organic matter entering the system in that along that coastal zone. So it's a pretty different looking setting than the areas that are mostly influenced by that those Pacific water masses that are moving north. So the result of these, this hydrography is a lot of spatial heterogeneity in the benthos across this system. And Jackie Brebmeyer, who's been working in this region since the 1980s, is a pretty well-known benthic ecologist. She has been sampling some of the same areas pretty much throughout her career and has trained others to help her in that effort. So a lot of these are her data from, these are only from the early 2000s, but her data goes back to the 80s. And what you see here is just benthic biomass and carbon units, and these regions that have been identified as what people are calling benthic hotspots. So places where there's a lot of particulate flux to the seafloor, partly because of some of these water mass features. And there are places that we know there to be elevated levels of benthic biomass. And there are also areas where we see intensive feeding activities of some of the benthic feeding mammal and bird species that are particularly of subsistence interest. So I'm going to talk a little bit about this region here in the black box, and that is the Southeast Chukchi biomass hotspot. And it's a place where there's an eddy feature that forms because of that constriction of flow through Bering Strait. And it's basically like a vortex that dumps particulate material onto the seafloor. So clams make up a lot of that biomass in those spots, and there are places where things like walrus that are benthic feeding marine mammals are foraging. Some of the other spots here have other dominant taxa, like there's a gray whale feeding area here that's mostly an amphipod zone. So the biomass is not the same things everywhere in those spots. So some of the long-term, one other point I wanted to make really quick. Anyway, the black boxes on that map represent the sites that are part of the Distributed Biological Observatory, which Jackie and others have established in this region. There are other distributed biological observatories elsewhere in the Arctic, but these are places where we have long-term data sets, and there's sort of a community-wide effort to try to visit those spots repeatedly. So when people are in the area for cruises, there's sort of a gentleman's agreement that they'll go to those spots and try to repeat some of these standardized measurements that are being made there. So just as an example of some of the trends that we're seeing from looking at some of these long-term data sets, first of all, some troubling changes in some commercially valuable species like snow crab. We had a huge snow crab crash in the fishery in the last couple of years. There was a heat wave in 2018. Snow crab didn't really come back from that. So to the point where there's been federal disaster relief funds allocated to the snow crab fishermen in response to that. And the current information seems to point to starvation conditions for snow crab in a key part of their range. But another maybe less, I don't know, charismatic faunal change that we're also seeing comes from Jackie's data, where she's seeing trends in certain places where there's shifts in, even though there are still bivalves there, which are some of the dominant fauna, there's shifts in what kinds of bivalves are there. Well, that doesn't seem like an obvious issue. If you're, say, a diving sea duck and you don't like this bivalve, you just like this one, then these are troubling changes for some of those taxa that actually feed benthically. And I will argue that they also seem to suggest some changes that are happening in terms of the metabolism of the benthic community. So one of my students did some incubation experiments with individual organisms in closed system respirometry experiments that we did on board a ship. And so her data are shown here. Each one of these colored lines represents a different bivalve species. And there's also an amphipod in here in the blue. So what you're looking at here is the mass specific oxygen consumption of individuals of these different taxa. And each dot is an individual that was incubated. So a couple of things to note. First of all, the magnitude of the respiration is different depending on the different species. So even for the lines that are parallel and are showing kind of the same mass to oxygen consumption relationship, there are differences, roughly an 8-fold variation among these species in terms of the respiration rate. So shifting from one species of bivalve to another has implications for how much carbon is being utilized by these organisms, which kind of dominate the biomass. So something like an order of magnitude range among those species in terms of carbon consumption is what we estimated. Also, there's a scaling law in metabolism that suggests that all organisms should exhibit more or less the same relationship between weight and oxygen consumption. And you could, like, we have a sense that like hummingbird metabolism is very high relative to something like elephant. So so 4 of the species that have the parallel lines here conform to that metabolic scaling law pretty well. A couple of them don't conform at all. So there are likely issues with variation here that's related to life stages. It's really difficult to sample larger individuals, older individuals of some of the really large clam species that are of particular importance in understanding the diet of some of those top predators. You just don't get them because they're so deep in the sediments. But there's also probably some relationship to feeding modes and lifestyle. So Macoma, for example, in the black line there, which shows the weirdest relationship, switches between a deposit feeder and a suspension feeder opportunistically. So it could be something to do with how Macoma is feeding. We don't really know. So I just show this data to emphasize that even though we might just be measuring, I might think clam biomass in and of itself is a useful measure. There's a lot of story here, thinking about which clams there are, and even going beyond that, the relative proportion of whatever clams to other things in this system. But we can't measure individual respiration rates on every single species. That would be kind of difficult to achieve. Maybe we'll get there someday. So I've really been thinking about how we can better generalize information that we get from looking at the fauna to broader areas. So this is an example from an interdisciplinary field project that we were involved in. And we had information about the macrofaunal community across this portion of the study area. So we had pretty good taxonomic information as well as a lot of other environmental parameters. And we also did flux core incubation experiments. So we have some information about oxygen and nutrient fluxes. And what you see here is the results of a multivariate community structure analysis, pretty standard thing in benthic ecology, where when we looked at specifically polykey functional groups that are widely described in the literature and include information about feeding mode and motility, the type of feeding structure that the polychaetes have and so on. We found that there were places where there were essentially the same community over a fairly broad area. This information was lost when we looked at higher level taxonomic groupings. So there's plants everywhere, there's polychaetes everywhere, there's amphipods everywhere. But when we got to the level of functional grouping, which doesn't even require a species level identification for polychaetes, we were able to kind of generalize patterns across this broad area. So the colors represent places where a specific community was found, where we had related taxonomic or functional relatedness within that grouping. Relationships to environmental variables that were most important were grain size, of course, but also bottom current speed, which we got from ADCP data from when we were underway on the ship, but also particulate nitrogen and salinity, which is probably telling us something again about the influence of that pressure water current along the coastline there. So I'm going to talk a little bit about the red versus the black stations in the second here. But I also want to note that these same spatial patterns were also observed in looking at the nematode communities or the myofaunal size fraction, and also in prokaryote community structure. So everything seemed to be consistent in terms of spatial patterns across this area. The microbial community structure was looked at by one of my other PhD students using 16S sequencing. And so we were able to infer some information based on taxonomy about what different groups of microbes were doing, but that's not straightforward to try to do. So this is, again, just throwing all of the species, not knowing or taxa, not knowing what all of them do into a multivariate analysis and looking to see where similar communities were found. So the colors in this figure represent similar communities in that sample. And these are depth layered samples at most of those stations that were shown on the map. So the stations are grouped into groups where we found communities that were kind of similarly distributed within the sediment. So without getting too far into the details, the red assemblage is indicative of anaerobic conditions. We have this surface assemblage that's found in a couple of different parts of the study region. We have the biomass hotspot area where we know there's a lot of bioturbation and a much more homogenized upper several centimeters of the sediment column with a fairly uniform assemblage of microbes. So it seems again like communities are reflecting modern spatial scale patterns over this region. As a side note, there are also cool things in the bacterial data that show that point to particular functions. Like we found abundance of things that are supposedly oil degraders concentrated in some areas. There's no visible evidence of actual oil there, so it could be that these are they're responding to alkanes and alkenes that are breakdown products of a lot of phytoplankton derived detritus, for example. And also, if we just look at surface sediments, microbial diversity data, again, over a much broader area that goes up into the Beaufort Sea, we see what I think is a surprising amount of consistency in the composition of the bacterial community over pretty large areas. So the Chukchi Sea is distinct from the Beaufort Sea for sure, but even over large areas in the Chukchi and even the northern Bering Sea, we see pretty similar communities. And then in the Beaufort, there's a lot of structuring between the nearshore and offshore. There's some other samples over here in purple that are probably distinct because of influence of the Mackenzie River, which is, I think, the largest volume river entering the Arctic, or one of the largest, carries a ton of organic matter into the nearshore environment with it. So that community looks distinct in bacteria as well as many other things. So again, I think that particularly as much as microbes reflect what's going on in the environment, this kind of tells us that there are generalizations that we can make over fairly broad areas about the factors that are important in structuring communities. and probably processes as well. So we threw together a lot of the information that we got from environmental variables and these different levels of the community as well. And there's a ton going on here that I don't want to get too bogged down in. I just want to make the point that it's possible to compile information about systems like this and set up some hypotheses that we can potentially test with models or vice versa. The black and the red here are, again, the contrasting conditions between that coastal current influence area versus the EVO3 hotspot area where we know there's a lot of deposition. We see differences just visually in terms of where the redox boundary appears to be, but also the bacteria kind of reflect what we just see by looking at the pores. So just an example of how we're kind of attempting to synthesize all this information into something that sort of divide the habitat up into what we kind of call ecoregions, where we see similar conditions, similar fauna, probably similar processes. So I'm going to focus for a second on the 3 stations that are in the little red box up there. So the 2 stations in red are kind of out in that biomass hotspot again, and then the 3rd station closest to the coastline is that coastal water mass. So just looking at some sort of bulk parameters, I guess, of the community structure. On the upper left here is just biomass distribution between macrofauna and bacteria. You can see, of course, the presence of those large bioturbating clam species offshore versus when you get into the coastal water mass, it's a lot more small-bodied, myfaunal sized organisms, small deposit feeding polychaetes and so on. So the gradient from sort of offshore to coastal regions looks something like this, where we have different bioturbation functional groups in the nearshore environment than we do in the offshore environment. Some other stuff going on in the middle, more amphipods, lots of amphipod diversity for some reason. So major functional differences in terms of the communities that reflect these gradients in the environment. So I guess my goal is to kind of think about what functions matter and what we can infer process-wise by looking at what we know about the fauna, because we know there's quite a bit of data about the fauna and about things like grain size across this area. So results of the oxygen flux experiments from those same 3 sites, which we conducted at 2 different temperatures. On the left is the total oxygen uptake from incubated pores at a couple of different temperatures, incubation temperatures. So you can clearly see the role that the big macrofaunal species are playing, like the bivalves in those places where they're more concentrated. But when you look at this information normalized to the biomass of the macrofauna, you see that pound for pound, those smaller body taxa in the coastal water masses are actually, I guess, more efficient or more rapidly processing that material. So I guess I interpret this as, I mean, it makes sense that there's a relationship between mass and metabolism to some extent, but I don't think the small fauna should be discounted from being important, and they often are kind of ignored in place of, just focusing on the large things that are sucking up a lot of the oxygen. Because the smaller fauna are turning over much more quickly. These are shorter lived species. And so here we might have carbon that's sequestered into bodies of clams that are living there for longer periods of time. But in the places where the smaller fauna dominate, there's a lot more turnover and higher secondary production, essentially, because there's just a lot more, the generation times are shorter, they're shorter lived species. So it could potentially just be more rapid cycling of that material. We also probably have the bacteria are suggesting that there's also a component of anaerobic activity happening more so in those coastal sites. And so some of this oxygen flux might be chemical uptake, but if you look at the ratio of DIC to oxygen consumption, which we would expect to be something like 1 for aerobic respiration, that fits pretty well in the places where we have big clams. Something else is going on in the middle here. And then in the coastal sites, we sometimes see, I mean, this ratio is still not that different than one, so I guess I would argue that it's not, that's not the whole story, it's not just anaerobic consumption of oxygen, that's different in that setting. We also did a pulse chase experiment on these at a couple of these sites with labeled, isotopically labeled private detritus. There's a ton of information to sift through in that data set, so we tracked it into the bacterial carbon pool and into all of the individual faunal components in the forms as well. So without, again, getting bogged too much down in the detail here, this is just an example of the type of information that we're getting from that type of studies. And this is families of polychaetes, and on the right, total consumption of that labeled isotope labeled detritus by individual taxa, mass specific uptake on the left. So one kind of contrasting or species or group that represents this contrast that I want to demonstrate is the spionids, which have a history of being highly particle selective. They're really good at selecting and finding the freshest, recently deposited private detritus particles. And they make up a pretty small component of the total biomass. So their peak here in terms of total uptake of that label is really small. But mass specific uptake is extremely high compared to everything else. And I think they're just really good at getting there first. And so even though they're not, their biomass is nowhere near that of clams, they are actually super efficient at processing that material. So this type of information about which fauna are there, even on a functional group level, could also give us some pretty useful information about the pathways that organic matter is being utilized and groups that might be using it more quickly than others in some places. So in trying to think about how to go from the information that we're collecting about individual metabolisms to stuff to making predictions on the broader scale about change in the system, I came across this idea, which I think is interesting, that talks about the metabolic niche of individual taxa, meaning that specifically certain species are adapted to a particular energy regime. And so particularly in this area, this paper is written in the context of the deep sea, where similar predictions have been made that the amount of flux is going to change through time with warming and climate change. That's the angle that they're coming from, but I think the same idea applies in my region, where the same predictions are made that we're going to see changes in either the quantity of flux or the nature of that material that's actually fluxing to the seafloor. So the energy availability is predicted to change going forward. So they say that species are adapted to these specific kind of energy conditions in their setting. And if we dampen the amount of flux to the seafloor, then we might expect those taxa that have high energy demands, like bivalves, for example, to be challenged in certain settings because they just require more food. So there's a couple of different hypotheses here about what we might expect to see with changes in flux, for example. And the axes are just the total metabolic demand on the y-axis, and then the chemical energy availability, the amount of food on the x-axis. So one hypothesis is that we'll just lose a lot of those high energy taxa from the system. And so in places where you have low energy availability, or where you have low metabolic demand, so the low energy taxa, they're fine no matter what, but the high energy demand taxa are eliminated from the system. And so they talk about how niche diversity basically should mirror what we see in terms of this alpha diversity or species diversity along that line. The other hypothesis is that there'll be competitive exclusion of the lower energy taxa. And so in this bottom example here, basically the low energy taxa are only found in low energy environments because they've been excluded in all other settings by the high energy demand taxa. And so that gives us a different shaped diversity curve, which looks a lot like the well described diversity curve that we see on continental slope environments, where you're going from a high productivity shelf environment to a low flux deep sea environment, and there's this sweet spot in the middle where there's overlap basically in the vertical ranges of those two different groups of organisms. All right, so I didn't want to, the other point that I think a lot about, and I didn't really have time to go into detail on, but I would like to throw it out as a thing to think about in our further discussions, is changes in the quality of the organic matter that we're likely to see. In my region, ice algae is a big part of the story. So as we lose sea ice, we lose ice algae. They have a different biochemical composition, partly because of how they grow in the ice matrix. We're also seeing and expecting to see further changes in the phytoplankton community structure. Phytoplankton also, individual species vary in terms of their nutritional value. They also vary in their nutritional value depending on their growth conditions. And then also terrestrial organic matter inputs are a big unknown in the system that I work in. We know there's a lot of it, but we don't know to what extent it actually enters the food web. And with increasing erosion and increasing runoff with melting glacier systems, for example, we're likely to see more of that material in some areas. So what does this mean? Does anybody care in the community? I think about all this stuff as food for animals. So there's a cool review paper that looks at quantity versus quality and what the links are between abundance or biomass of major groups of organisms, myofauna, macrofauna, megafauna, and shows that megafauna, for example, care more about quality or quantity. Smaller organisms like myofauna are more sensitive to changes in the composition of the organic matter, so total lipids or total labile protein, that sort of thing. So my general question, I guess, is what constitutes quality? How do we measure it? What matters for organisms? Certainly there's implications for the geochemistry of that material as well, the reactivity of that material. So just as a couple of examples, on the lower left here is a figure from a fairly recent paper that used an isotope labeling approach to look at breakdown, remineralization, and assimilation of ice algae and phytoplankton in sediments in the Arctic, and found that there was a different response to ice algal inputs versus phytoplankton, and that response was not consistent between the two stations that they looked at. So different communities responded slightly differently. On the right is another example from a grad student from my department who used isotope compound-specific amino acid isotope approaches to try to track bacteria, phytoplankton, and terrestrial organic matter into the benthic food web. And she asked the question of whether different feeding types use different components of those different components of the food source in different proportions. And she did not really find any patterns when she just looked at like average suspension feeders versus average deposit feeders, etc. But when you dig into groups or taxa within those categories, like on the bottom here, two different subsurface deposit feeders, they use that resource pool differently, even though they're both subsurface deposit feeders. So what is it about Maldanids that they particularly prefer one resource over the other? And again, like coming back to the question of what sort of information do we need to capture about the benthic community, that's informative to models. Like if we want to think about processing of these different organic matter sources, what sort of functional information is useful in doing that? So yeah, maybe. Anyways, yeah, so this is just a recap of some of the things I just said and things that I think that would be useful to know, more metabolic rate information for some of the key species. We don't know much about microbial biomass in sediments, especially in the Arctic, and how respiration is partitioned among the different components of the pond. Yeah, anyways.
Great, thanks. Next up, we have, Nick, right? He's talking about small scale. I lost it.
Yeah,
Sediment water interactions.
yeah, so the title I was given was small scale processes regulating sediment water interactions. And about halfway through making the talk, I realized that's not exactly what I was making the talk about. And so I'll start very quickly talking about small scale sediment water interactions, but I'm just going to talk about what is small scale, when does small matter, how might we think about small at an ecosystem level in models. And so I think if you're thinking about small scale sediment water interaction processes, this might be kind of a perfect picture to look through and kind of try and pick out some of these interactions. So it's kind of late in the afternoon, so do some audience interaction. You can see something up here. I'll throw you some candy. Some candy. So who can see something that looks like a small scale sediment water interaction? There's at least 3 or 4 things that might matter biogeochemically.
Bed forms.
Bed forms, right? We have some ripples in the sand. What else? What else do we have?
There's bumpy things in the little lines.
There's bumpy things in the little lines, right? All right, so we have these bumpy things.
Yeah.
Right, so we can see there's different places where organic matter is accumulating, right? And there's some places with no organic matter. We're just gonna assume that's organic matter, right? What else? There's at least 2 more things.
Bits and pieces of detritus.
Okay, bits and pieces of detritus, so hot spots.
Yeah, sure.
Sorry.
I got it.
All right, there's 2 more things I see. We'll go one or 2 more audience interaction, then I'll just say. them.
Small scale current variation across the trough and the crest.
Yeah, so we have these different things, so like maybe differences in turbulence going on in there. All right, one more thing here that maybe is not so important. We're moving out to sea. And that's just, I see it as there's water here, there's no water here, right? So maybe we have some different saturation states. Okay, so we can think about all these small scale processes. They change oxygen conditions, they're going to change our diffusion, they're going to change how organic matter can move around in this system, how electrons can move around in this system. They're going to turn on and off different groups of microbes. Maybe we also see, if we look super closely, there's like a little shell down here, right? So there's probably some bioturbators in here. So you can think about all of these things, and they matter in like the square meter, or maybe they matter in like your sediment core. But how do we get these up to ecosystem scale? And when I was told I was talking about small scale processes, I think about small scale at like the ecosystem scale. So what is small when you're trying to think about a big system? And so to give some ideas about how I think about this, so I might think about what happens if we put an oyster reef in an estuary, right? How does that change the nitrogen cycle of the system? I've also done some work thinking about ponds in the landscape. So ponds, like the little bodies of water less than a hectare, are responsible for more than 5% of all methane in the atmosphere, all methane released to the atmosphere each year. We also just wrapped up kind of a meta-analysis machine learning exercise where we also estimate that ponds bury more carbon than lakes and reservoirs globally. And then we can also start thinking even bigger scale. We can say, what happens with coastal erosion? How does coastal erosion interact with continental shelf biogeochemistry? So the example I'm showing here is kind of coastal permafrost or eudoma collapse in the Arctic, and thinking about what does that mean for the carbon or the silicon cycling out on this continental shelf. And basically here we can estimate that coastal erosion maybe is loading as much particulate silicon as rivers are to the Arctic Ocean. So it's pretty important, but it's happening in this really small space. And so the question really becomes like, who cares? When do small scale processes matter and what is small scale? And so I tried to track down a definition of like, what is small scale? That's a really hard definition to find. But I did remember a quote from Bill Schlesinger from a talk he gave. And he basically said, this is how I remember it, so it's not exactly correct. So he said, something is biogeochemically important if it makes up like 3% or less of the space or mass of a system, but is responsible for 10% of a process flux or pool, right? So this is how maybe we can think about or start to try to define what is a small scale process that matters biogeochemically, right? Maybe we can make a very simple little graph like this, right? The X and Y axis are not the same here, right? And so I thought it would be fun to revisit some of my dissertation research and ask, do oysters matter? And does putting an oyster reef matter in the context of like this small scale process in a bigger system? Did my dissertation research matter? Right? And so to kind of give an overview really quickly of how I'm thinking about ecosystems and carbon and sediments. An oyster reef, you can imagine the oysters are sitting here, filter feeding out particulates as they go past, loading most of this organic material to the sediments, kind of changing the biogeochemical conditions there. But they're also changing some of the currents. We're going to bury some shells, some calcium carbonate. We have this other habitat going on where we might be providing surface area for macroalgae to grow, for other fish and crabs to grow, and we're going to have going to see a change in carbon flux back to the water column, but we're also going to see a change in burial of particulate carbon. And so a lot of my dissertation research was spent out in oyster farms like this. Each of these bags is holding about 150 or 200 adult oysters. And the data I'm about to show on the next slide is just comparing sediment flux of methane between the sediment and the water column in these bags, and the same measurements about 10 or 20 meters away. All right. And so we did this at several different locations. These are all in New England. So this is Duxbury Bay. So this is in coastal Massachusetts. So we have a really high tidal range. Sediments are pretty, almost like sticky, kind of clayey. The tidal range is about 2.5 to 3 meters a day, and the whole bay drains, right? And if we look at methane flux between sediments without oysters, our control site, and then with oysters, we see, look, okay, maybe we're having a bit more methane production. If we go to kind of the opposite conditions, so Ninigret Pond, this is a coastal lagoon in Rhode Island. It's microtidal. There's not a lot of wave energy going on. It's pretty sandy and low, relatively low organic matter content. We see that our control site is much lower than in Duxbury. But we do still see this effect of oysters there, right? But putting more organic material into the sediments, we see more methane. We see the opposite effect of oysters at Roger Williams. And so this is Roger Williams University in Narragansett Bay has a little pilot scale oyster farm. They also have an oyster hatchery, but this is really kind of gravelly, sandy springs with a lot of wave energy. But a 5-minute boat ride away, there's a small lagoon with a commercial oyster farm with really sulfidic mucky sediments and really low wave energy. And we see that we have a lot more methane production, but no effect of putting this small scale process there, right? And so some of the work we were doing was trying to dig in and think about this on a site specific level and think about the mechanisms within a site, right? And so we were starting to think about biodeposition, so how much of the particulates in the water column were oysters, filter feeding and loading sediments based on abundance, and then how does that influence rate? We're thinking about what else is living there, what about competition for food between the oysters and these other organisms living there, and what does that do to the carbon respiration, right? So maybe we have this spot where there's no food, so we don't really have much organic matter loading, then this sweet spot where there's plenty of food, plenty of waste that can drive methane production. And then at the end here, we have so much competition for food that we're reusing waste as a resource, right? And there's not a lot of organic material available. But we can start digging in and doing this and start trying to hypothesize mechanisms to test. But it maybe matters more to think about in an ecosystem context, does this matter, right? Do oysters and does oyster mediated methane cycling have an impact at the ecosystem scale? So I just put together a really simple kind of scenario thought experiment. We're gonna have 4 scenarios, 100 square kilometers. We're gonna assume these sediments are perfectly homogeneous. You know, totally bare sediment, or mostly bare sediment with our small scale process there, that if it makes a 10% difference in the biogeochemistry of the system, it's important. We're also just gonna test some seagrass, and then we're gonna kind of have this little mosaic habitat, right? Where we have oysters and seagrass and bare sediment. And the reason I think this is kind of a useful little experiment is because it can bring us to some important questions for thinking about how to think about and include small scale processes and models. And I'm talking about this kind of in this ecosystem context, but these same questions will apply to a smaller, like a meter square estimate. And so we're going to make some very, very simple assumptions. We're going to think about carbon stocks. We're going to think about carbon fluxes, all right? And so we're going to assume seagrass carbon stocks about 150 tons of carbon per hectare. Bare sediment, we're going to say 20. Aquaculture, we're going to say 30. Then for our fluxes, our seagrasses that are blue carbon systems that are sinking carbon. So we're going to say negative 20 kilograms of carbon per hectare per year. Bare sediments are going to be releasing 5 kilograms, and our aquaculture is going to release more. So we're going to say that we're our most extreme version of aquaculture that's pumping out carbon. And so if we look at our carbon stocks, aquaculture does not matter for carbon stocks, right? So it's really only going to be a 1.5% increase if we think about the whole system. But if we think about the carbon stock of this system in scenario 3, the small fraction, the seagrass that only covers 3% of the system, is responsible for 19% of the burial. So this small process, or the small piece of the landscape really matters, right? That makes sense. This is why people care about these blue carbon ecosystems. But then we can also go to this last one and look at, well, what happens if we have this mosaic? If we increase our seagrass, and we see that there's even more importance, that the oysters become even less important in that context. If we move and then start thinking about the fluxes, right? In our first scenario, we see that, yep, we're releasing carbon from the sediments of the water column. We just have aquaculture. We have 6% of emissions are attributable to this oyster farm. When we have seagrass, our carbon flux goes down. But then if we go over here to scenario 4, and this is where we start getting into this tricky space, we're trying to think about what small scale processes should we care about. Here, our oyster flux is 11% of this system-wide budget, right? And so if we think about this in the context of be 3% or less of the system, but make 10% or more of the process, oysters do matter. But then in a different context, they don't, right? And so this, I think, kind of brings up this interesting question that we can start to think about when we're trying to think about what small scale processes to include in models. And so that example I was just showing, measurements made right in that little system, or right where we had that different structure, this different habitat. But oysters and other organisms are also doing a lot of other things. Right? We're forming shell, and we're changing the carbonate cycle. We're interacting with the DFC pool through respiration, consumption, incorporation of carbonate tissue, or provisioning habitat for other organisms that are doing similar things. And I'm not going to get too much into this, but there was this really I found this really exciting study that came out last year, looking at how an oyster reef might control the carbonate chemistry in the ecosystem there. Right, and so kind of one of the most interesting figures from this study is this ambient seawater pH versus oyster reef pH. And what it shows is that if you're near an oyster reef, your pH goes down. Right? It's kind of interesting. And this can be attributed, and the authors attributed this directly to the deposition of oyster shell. Right, so the process of forming this oyster reef is acidifying the ecosystem around it. Right? And so we can think about this also, kind of processes like this, also in our kind of small scale, does it matter question, right? We have our bare sediment, our aquaculture, but then we can maybe say we're influencing a larger area, this area where we have perhaps this kind of effect in the water column of these oysters. And how does that change in pH and change in oxygen feedback to sediment processes? And so I think these are kind of important questions. Does this still count as a small scale process? Right? And I'm going to wrap up because I'm the last one between you and coffee. And I kind of have these four questions. When thinking about small scale processes, and trying to include small scale processes in biogeochemical models. And the first thing we need to think about is what is small scale? How do we define small? Right? The second, and I think this might be the most important question, is how important does this process need to be for inclusion in our model or for our scale, right? Is 3%, is 10%, like these are kind of arbitrary numbers, and we could argue about this all day. We also should think about how the process should be considered, right? We can think about bare sediments versus oysters, or bare sediments versus seagrass oysters, right? When this, whatever the importance of this oyster matters, should we think about just in that system itself, or kind of this cloud, or this space around the system, around in the system where the small scale process or habitat is influencing things. And then we also need to think about interactions between cycles, right? And so kind of the longer example I gave was thinking about carbon and methane, but we also might think about oysters are releasing nutrients and how does that feed back to the ecosystem, recycling nutrients and driving additional primary production, and kind of translocating where organic matter might be produced and deposited. So I think these are some fun questions we can talk about. This is how I think about small scale, not really that small. It's small in a big space. Yeah, I would love to talk about this. Thanks.